This is the cell-mediated immune response video. This video goes along with the worksheet in your course pack. Cytotoxic T cells are involved in the cell-mediated immune response. This type of response most commonly occurs with virally infected cells or cells with intracellular bacteria infections. For this video, we will use a virally infected cell as our example. So we'll start with a review of viral replication. In order for a virus to infect a cell, the host cell must have the appropriate receptor. The virus surface proteins are like little keys that attach to the host cell receptors, which would act like a lock. The virus can enter the cell through either endocytosis, for a naked or enveloped virus, or fusion with the cell membrane, which would only occur with envelope virus. Here we have one of your cells, and these little structures on the surface would be receptors. The shape of this receptor indicates what type of virus can attach to that receptor. So here's the dreaded coronavirus image you see all over the news these days. This virus has these protein spikes that allow it to attach to a receptor. Once it attaches, that virus is able to enter into the host. After the virus enters into the cell, the capsid is degraded and the virus exists only as the nucleic acid. The nucleic acid has the genetic code instructions that allow the virus to hijack the host cell enzymes in order to replicate. The viral nucleic acid is replicated and the viral genes undergo transcription, then translation, which allows the virus to be assembled. Transcription of the DNA or RNA would eventually produce the viral proteins. Those viral proteins are assembled into the virus, and that virus can then leave the host. The virus leaves the host through exocytosis, lysis, or budding, depending on the type of virus. While inside the cell, the virus is hidden from our immune system. The virally infected cell will process endogenous antigens, which are the intracellular antigens, and display them with MHC class I proteins. It will do this, package that protein with MHC1, and then moves it out onto the cell surface. This is a flag to our immune system. Our immune cells will then be able to search for these viral antigens. This is number 1 on the worksheet. An antigen-presenting cell, such as a dendritic cell, which remember are the scout cells, they're constantly sampling antigen. They recognize this endogenous antigen with MHC class I protein, and the dendritic cell takes the antigen to the lymph node. Next, we have antigen presentation to a cytotoxic T cell. Cytotoxic T cells have TCRs, or T cell receptors, for one antigen. They also have CD8 protein, which recognizes the MHC1 protein. These cytotoxic T cells are constantly surveying the antigen-presenting cells for their complementary antigen. The cytotoxic T cell with the correct T cell receptor will then be allowed to attach to that. The CD8 protein then recognizes MHC1 and stabilizes that bond between these two cells. It's important that the viral antigen is on that MHC1, so cytotoxic T cells have CD8 that only recognize endogenous antigen presented on that MHC1. Next, we have cytotoxic T cell activation and clonal expansion. Once the antigen presenting cell finds the correct cytotoxic T cell, a signal from a T helper cell is needed for activating the cytotoxic T cell. So here we have our signal to that cytotoxic T cell, and that signal causes clonal expansion. So we get more of those cytotoxic T cells produced. Some of these clones become memory T cells, which are involved in a faster secondary immune response. Activated cytotoxic T cells leave the lymph node and seek out virally infected cells with endogenous antigen packaged with MHC1. This is the cell from one of the first slides in the description of cell-mediated immune response. So this cytotoxic T cell finds this infected cell, and once it binds, it has its CD8 protein that stabilizes the bond between these two cells. Once bound, that cytotoxic T cell kills that target cell by secreting such factors such as perforin, which puts pores into the cell membrane, and then granzyme, and granzyme goes through that perforin pore and activates apoptosis, which kills that cell. This concludes the video for cell-mediated immune response.